In this lecture, we'll be covering the central dogma, some basics of genetics, and finishing up with microevolution. Our objectives today are to identify steps in the central dogma, to be able to define DNA, RNA, protein, mutation, allele, heterozygous, homozygous, dominant, recessive, segregation, independent assortment, and of course, evolution, and to explain how different alleles are formed. A lot of this will be reviewed if you took Bio141. So what is the central dogma? The central dogma is the basic idea of how genes are inherited and how genes are expressed. So let's look at a gene here. A gene is a segment of DNA that is going to be expressed as a produced protein. As you see, here you have your segment of DNA there with associated histone proteins, a promoter region, two enhancer regions. This will come up later in genetics and cell biology, but for now, let's focus on what that gene does. It is going to be read as DNA triplets. So DNA is read in groups of 3, and we're going to go through that quite a bit today. And it's read by transcribing it into mRNA, or messenger RNA. This messenger RNA will leave the nucleus and will be read by a ribosome and translated into an amino acid sequence. Each DNA triplet is going to code for an mRNA codon, which is then going to code for a single amino acid. Those amino acids will be bound together with peptide bonds and will form a functional protein. So how are these instructions encoded into DNA? Well, there are 20 amino acids, but there are only 4 nucleotide bases. So how do we make a code? Well, you look at this and you say, okay, how many nucleotides in a row would be needed to code for a single amino acid so that we have 20? If it was 1 amino acid for 1 nucleotide base, well, then you can code for 4. If it was in pairs, 16. If it was in triplets, 64. Well, 64 is larger than 16, but it leaves a lot of different codons that can all code for the same protein. We say the protein code is degenerate. What this means, though, is that mutations in the DNA can sometimes code for the same amino acid. And let's look at that DNA code right there. You do not have to memorize for this class, or probably ever. But you see there are multiple stop codons. There are multiple codons for phenylalanine. Leucine has 6. And this means that you can have mutations that do not cause a change in amino acid. What types of mutations do we have? Well, we have gene-level mutations, which you can have things like point mutations, which are the addition, deletion, or substitution of a single nucleotide. As I said before, the substitution may or may not lead to a different amino acid when transcription and translation are done. Addition and deletion can cause their own problems. You can have short tandem repeats, which add a small number of nucleotide, which is going to duplicate those small numbers of nucleotides again and again, and that can actually lead to the same amino acid being repeated several times in a protein. Or we can have, of course, whole chromosome level mutations, such as duplications, deletions, and rearrangements of relatively large sections of DNA. We're not going to look a lot into those until we get into some later genetic stuff, but this can cause changes in how often genes cross over during prophase 1 of meiosis. So duplications, deletions, or flipping or moving to the other parts of the chromosome, these are all different types of chromosome level mutations. So we've been saying the word allele a lot. What is an allele? An allele is a variant of a gene. And that can mean that one gene can have several possible different sequences based on mutations in some organisms, but not others. So thinking about this to the organismal side, an organism can be homozygous if it has 2 copies of the gene, and both of them are read the exact same. An organism can be heterozygous if it has 2 copies of the gene, but they have different alleles because they have different sequences. And as we saw earlier, different sequences can lead to different amino acids, which can lead to different proteins, and different proteins can cause different functions in the organism. Here we have one example of a gene. This is for a gibberellic acid protein, and we'll get into that in lab. If you have the functional copy, this makes the pea plant tall. If you have a non-functional copy due to a mutation that makes a non-functional protein, that makes the plant a dwarf. These are two different alleles of the same gene due to different nucleotide sequences that cause different proteins. So let's look back at meiosis and look at some basic laws of genetics as a refresher. First off, the law of segregation. When gametes are formed during meiosis, your homologous chromosomes are going to separate from one another, and they're going to go into separate gametes. This means alleles are going to segregate, and each gamete is going to receive but a single allele for each gene. We see that here in meiosis 1, the homologs separate, and as they separate, you have the segregation of all the tall alleles into one daughter cell and all the dwarf alleles into the other daughter cell. In meiosis 2, the sister chromatids are going to separate, and that is going to mean that there is only a single copy of each gene in each gamete. Next, the law of dominance. If an organism does have two different alleles, but you only observe one trait, that is the phenotype. That means the dominant allele. The other allele is recessive. So, what's the difference here between phenotype and genotype? An organism can have two functional copies for the tall allele, and that'll make a tall plant. If an organism has one functional copy of the tall allele, it still has a functional copy, and that makes it a tall plant. Both of these plants have the same phenotype. They are tall. They have different genotypes. One is homozygous, the other is heterozygous. This answers the question if organisms can have the same phenotype but different genotypes with a simple yes. Likewise, for purple flowers or white flowers, if an organism has two copies of the dominant allele, they're homozygous dominant, producing purple flowers. If they have only one copy, they can be heterozygous. If they have both copies of the non-functional allele, they're homozygous recessive. The law of independent assortment is that each pair of alleles is going to segregate independently of other pairs of alleles. This is, of course, considering that genes are not right next to each other on the chromosome, which is called genetic linkage. We'll get into that in another class. But law of independent assortment means you get different combinations of two traits because of how the homologs align in metaphase 1. See here that you have two pairs of homologs, and the maternal set of chromosomes can align where both of them are on the right with the paternal on the left. Or they can mix it up a little bit. These are equally probable possibilities, and during meiosis 1, they're going to separate into the different daughter cells, and they can separate differently. They can assort independently of one another, causing different combinations of chromosomes. You have 4 possible combinations coming off of these two homologs. So this is a tracking question for in class. You might want to check yourself and see if you are able to explain this. Let's look at Punnett squares a little now. So let's say you have two organisms. They're both heterozygous. We're going to cross them, and you can see that there are different possibilities of what we're going to get as offspring. But we're also going to see that there are probabilities. In two heterozygous organisms, we see the probability is 50-50 for each one of the different alleles. And what we're going to see is the multiplication of 0.5 times 0.5 leads to a 0.25 probability of each different genotype. Now you see that the heterozygous genotype, there are 2 ways to get it. We would add those 2 heterozygotes together and say there's a 50% chance of producing a heterozygote. This is in 2 individuals that are mating, and these are the probabilities. Next time we are going to cover what happens when a gene pool is all mixed together and the resultant probabilities of that. What happens when the frequency of alleles change, though? So we're going to look at gene pools next time and look at how the frequency of these different alleles will change in a population over generations, and that is going to be evolution. We're going to see the change in allele frequencies resulting in organisms looking different over time, as we see with these dog breeds. And our focus in the next couple of lectures is going to be on allele frequencies, populations, and looking over generations.